Hello friends, welcome you all. I am Dr. Kalim Sheikh, Assistant Professor in Zoology, Pune College, Pune. Today we are going to talk about the chemical nature of cell membrane. Friends, do you know the cell is basic structural and functional unit of life? It is very complex structure, mainly consists of cell membrane, cytoplasm and nucleus. The cell membrane is the edge of life. It is the boundary that separates the living cell from its surroundings. A remarkable film, only about 8 nanometer thick, it would be take over 8,000 plasma membranes to equal the thickness of a page of book. The plasma membrane controls traffic into and out of the cell it surrounds. Like all biological membranes, the cell membrane exhibits selective permeability, that is, it allows some substances to cross it more easily than other substances. One of the earliest episodes in the evolution of life may have been the formation of a membrane that enclosed a solution different from the surrounding solution, while still permitting the uptake of nutrients and elimination of waste products. The ability of the cell to discriminate in its chemical exchange with its environment is fundamental to life, and it is the cell membrane and its component molecules that make this selectivity possible. So, in this lecture, we will explore more details about chemistry of cell membrane to understand chemical components that make up membrane and their importance for the membrane functions. Cell membrane is a complex dynamic structure basically made of lipids, protein molecules that are in constant motion. The cell membrane is also called cytoplasmic membrane, plasma membrane or plasmalemma. The term cell membrane was coined by Nageli and Karmer in 1855, and the term plasmalemma has been given by Plough in 1931. Chemically, cell membrane and other membranes of different organelles are found to contain proteins, lipids, and carbohydrates, but in different ratios. For example, in the plasma membrane of human red blood cells, proteins represent 52%, lipids 40%, and carbohydrates 8%. Now, first of all, see about the membrane lipids. Lipids are essential for membranes because they organize in lipid bilayers, that is, cellular membranes, and they can modulate membrane properties in many ways. Since there are more than 1000 different lipid species in eukaryotic membranes, the four major classes of lipids are commonly present in the plasma membrane, and other membranes such as phospholipids, which are most abundant lipid in cell membrane, sphingolipids, glycolipids, and sterols. The sterols are in the form of cholesterol. All of them are amphipathic molecules, means possessing both hydrophilic and hydrophobic domains. Cholesterol is especially abundant in the plasma membrane of mammalian cells and absent from prokaryotic cells. Cardiolipin is restricted to the inner mitochondrial membrane. Now see about structure of the membrane lipids. The most common type of membrane lipid is the phospholipid. See in the figure, one phospholipids arranged in a bilayer form and make up the basic fabric of the plasma membrane. They are well suited for this role because they are amphipathic, means they have both hydrophilic and hydrophobic regions. In the figure 2, a single phospholipid molecule is magnified. From this, we can understand the actual structure of lipid molecule. See in this figure, there are two different domains in a phospholipid molecule, such as hydrophilic and hydrophobic. The hydrophilic or water-loving portion of a phospholipid is called as head, which contains a negatively charged phosphate group as well as an additional small group indicated as capital R in the diagram at left side, which may also or be charged or polar in nature. The hydrophilic heads of the phospholipids in a membrane bilayer face outward, contacting the aqueous or the watery fluid both inside and outside of the cell. Since water is a polar molecule, it readily forms electrostatic, means charge-based interactions with the phospholipid heads. The other parts of phospholipid molecule is the hydrophobic or the water-fearing in nature. It consists saturated and unsaturated long non-polar fatty acids called tails. The fatty acid tails can easily interact with other non-polar molecules but they interact poorly with water. Because of this, it is more energetically favorable for the phospholipids to tuck their fatty acid tails away in the interior of the membrane, where they are sheltered from the surrounding water. The phospholipid bilayer formed by these interactions makes a good barrier between the interior and exterior of the cell. Because water and other polar or charged substances cannot easily cross the hydrophobic core of the membrane. Let us see about functions of lipids in cell membrane. Membrane lipids are a class of molecules that play a critical role in the formation of structure with the association of proteins of the cell membrane. The fluidity of the lipid bilayer permits for flexibility and dynamic changes in membrane shape, and it is essential for various cellular processes. Membrane lipids create the foundation of cell membranes, enabling compartmentalization, protection, and selective transport of molecules in and out of the cell. Lipids also regulates the permeability of the membrane. Okay, now see the second vital component of the cell membrane is proteins. The cell membrane protein play very crucial roles in the structure and function of cell membranes. These proteins are embedded within or associated with the lipid bilayer of the cell membrane and perform a wide range of functions. Here we are discussing some important types of membrane proteins and their functions. According to their position in the cell membrane, the proteins fall into two main types: the integral or intrinsic proteins and the peripheral or extrinsic proteins, both of which may be either ectoproteins, laying or exposing to external or extracytoplasmic surface of the membrane, or endoproteins, laying or sticking out at the inner or cytoplasmic surface of the plasma membrane. The intrinsic proteins tend to associate firmly with the membrane, while the extrinsic proteins have weaker association and are bound to lipids of membrane by electrostatic interaction. Integral membrane proteins are classified into two categories, such as transmembrane proteins and lipid anchored proteins. The transmembrane proteins span the membrane completely with portion exposed on both sides. They often have hydrophobic segments that anchor them within the lipid bilayer. For example, ion channels, transporters, and receptors. Lipid anchored proteins. These are the anchored to the membrane via covalently attached lipids. They are important for signaling and cell recognition. The another type of protein is the peripheral membrane proteins. They are not embedded within the lipid bilayer but are associated with the membrane, often through electrostatic interactions with integral membrane protein or lipids. Peripheral membrane proteins are highly unique amphipathic proteins that interact with the membrane indirectly using electrostatic or the hydrophobic interactions, or directly using hydrophobic tails. In addition to their unique relationship with the cell membrane, peripheral membrane proteins often play a key role in the development of human diseases such as African sleeping sickness, cancer, and atherosclerosis. The study of these proteins will give new insights into their function and structure and may ultimately lead to groundbreaking advances in the treatment of severe diseases. They can be involved in signaling, structural support, and other functions. Now we discuss some membrane proteins on the basis of their function in the cell. The glycoproteins. Many membrane proteins have a carbohydrate chains. Attached to them are the glycoproteins. They can operate in the aqueous environment due to their hydrophilic character, which play a role in chemical bonding and cell-to-cell recognition. They enable cells to recognize another cell as a familiar or the foreign, which is called cell-to-cell recognition. They also help cell attach to and bind other cells, which is called cell adhesion. They play various roles in our body, including those related to our immune systems, physical protection, cell-to-cell communication, and reproductive systems. The another protein is the ion channel proteins. These are integral membrane proteins that form channels or pores and allowing the selective passage of ions, that is sodium, potassium, and calcium ions, across the cell membrane. They are critical for electrical signaling in neurons and muscle cells. The example of ion channel protein is a sodium potassium pump. This pump helps to exchange the sodium and potassium ions across the cell membrane during active transportation. The receptor protein. Membranes have surface receptors that recognize signaling molecules such as peptide hormones. Binding of the signal activates other proteins to a process called signal transduction. Cell membrane receptors bind to specific ligands, that is hormones, neurotransmitters, and transmit signals into the cell, initiating various cellular responses. One of the common type of signal transduction is mediated by a class of proteins called G proteins. For example, a hormone travels through the circulatory system to cells throughout the body. Now we can understand this signal transduction with an example. When a hormone finds specific receptor protein, it binds to the extracellular site of receptor, causing a conformational changes in receptor protein that affects its intracellular shape. Then the G protein diffuses laterally along the membrane and binds to the activated hormone receptor. This event triggers an exchange of GDP for a GTP molecule bound to the G protein. Now G protein diffuses laterally along the membrane and binds to an inactive enzyme. The terminal phosphate group of GTP is transferred to the enzyme and forming a phosphorylated enzyme. This event causes shape change in the active site, making the enzyme catalytically active. This active enzyme is now able to catalyze a reaction. The carrier proteins. These proteins facilitate the movement of specific molecules across the membrane. For example, glucose transporters, which helps transport glucose into cell. Glucose transporters, proteins, accomplish the movement of glucose from the extracellular space deriving from the bloodstream into cells. The transportation of glucose molecules in the cells with the help of carrier proteins present in cell membrane during facilitative diffusion. Now we can understand this interesting mechanism with help of the figure A. So you concentrate on figure A. See, the step 1, in this glucose binds to a GLUT1 transporter protein that has its binding site open to the outside of cell, that is T1 conformation. See the step 2, in this step glucose binding causes the GLUT1 transporter to shift to its T2 conformation with the binding site open to the inside of the cell. In the step 3, in this step glucose is released to interior of the cell and initiating the second conformational change in the GLUT1 transporter. In the last step here, due to loss of bound glucose, the GLUT1 transporter protein return to its original T1 conformational stage and ready for the next transport cycle. In this way, the glucose molecules are transported from extracellular space to intracellular space with the help of carrier or transporter proteins present in cell membrane during the facilitative diffusion transport process. The next membrane protein is structural proteins. Some membrane proteins provide structural support to the cell membrane. For example, spectrin in red blood cells and dystrophin in the muscle cells. Now, see the enzyme protein. These proteins are associated with the cell membrane and are involved in various metabolic pathways. For example, adenylyl cyclase catalyzes the production of cyclic adenosine monophosphate in response to extracellular signals. The third important component of the plasma membrane is a carbohydrates. See in the figure 1, they are always found on the exterior surface of cells and are bound either to proteins forming glycoproteins or to lipids forming glycolipids. They are present as short, unbranched or branched chains of sugars, that is oligosaccharides, attached either to exterior ectoproteins forming glycoproteins or to the polar end of phospholipids at the external surface of the plasma membrane forming glycolipids. No carbohydrate is located at the cytoplasmic or inner surface of the plasma membrane. All types of oligosaccharides of the plasma membrane are formed by various combinations of 6 principal sugars, all of which are glucose derivatives: D-galactose, D-mannose, L-fructose, salicylic acid, N-acetyl-D-glucosamine, and N-acetyl-D-galactosamine. Glycoprotein and glycolipids patterns on the surface of cells give many viruses an opportunity for infection. HIV and hepatitis viruses infect only specific organs or cells in the human body. HIV is able to penetrate the plasma membrane of a subtype of lymphocytes called T helper cells, as well as some monocytes and central nervous system cells. So friends, in this lecture, we understand the chemical nature of cell membrane, various components of membrane and their functions in brief. I hope this lecture would be enhance your knowledge and create interest to study the other important aspects related to cell membrane. Thank you.